Hello, my name is Amy Mason. I work at the University of Cambridge and I work in the Cardiovascular Epidemiology unit. As the name suggests, I work primarily on heart diseases. When we think about heart disease, we are often thinking about what drugs could we give people to improve their lives, or what interventions could we give people to improve their lives? And the sort of gold standard of whether or not something works is a randomized controlled trial. So we give half the people a drug and half the people no drug, and we see, okay, how well do they do some years down the line? And if we see that, on average, the people with the drug are doing far better, then we say, excellent, this is a good drug, we should give it to more people. But there's a downside to this too. Trials are expensive. Trials are time consuming. They take literally years to be able to follow people long enough to see these events. And they're difficult for the people in the trial too. They have to, forego the opportunity to try other treatments for their condition. So there's a possibility that their lives may be made worse off by being in the trial, which isn't great. It'd be really nice if we could do trials without involving people. And I work in an area where we try and use genetics to mimic a trial using Mendelian randomization. So the idea here is instead of a drug, we look at DNA. And we think of DNA being very randomized around the population. And we can look at all the people who have a particular variation called a SNP in their DNA. And if we could find a SNP that changes the protein we're interested in, for example, lipoprotein A, we could look at how that impacts them throughout their lives. So if we could find such a SNP, something that increasing the amount of that SNP that you have in your DNA increases the amount of lipoprotein A that you have, increases the amount of heart disease that occurs in people's lives, well, then we'd be like, okay, it seems like changing lipoprotein A does cause a change in heart disease. And we can think of this just like the randomized trial, that, half the people have the DNA, half of them don't. How does this impact them over time? And hopefully, we can find a detectable difference and think about how that then impacts the rest of the population. But we might be thinking, okay, one DNA change, maybe it's just coincidence. What if that DNA change is doing other things in the body? It's like, okay, that is a fair concern. But we don't just look at one, we look at hundreds. We find lots of different variants that change lipoprotein A and look at their impact in heart disease over a person's life. And if you look at this graph, you'll see that there's a very subtle trend up from the bottom left to the top right, suggesting that, okay, every time a genetic variant is changing lipoprotein A, it's also changing heart disease. We start to be very convinced that, yes, this is something that is causing it. And even more than that, we can get an estimate on how much you'd have to change lipoprotein A in order to be able to make the difference in heart disease detectable in, say, a 5-year trial. So when we did this at Cambridge, we found that you'd have to lower the concentration by about 100 milligrams per deciliter to be able to find a detectable change. But most people have far lower amounts than that in their blood. So if we want to test this as a drug, we'd be much better off at targeting people with very, very high concentrations of Lp(a) and really dramatically lowering that. So that means that hopefully we've managed to save a bunch of people having to be in a trial that wasn't useful, and potentially save the trial from turning out, no results because it was testing the drug on the wrong people. And that's how we can use our research to help improve drug trials. Thank you very much for listening to me. I hope you found it interesting.